Yeah, thank you. So as Pia said, I'll be your first presenter right now. I'm covering for my colleague, but I will make sure that everything she wanted to tell you is said in this webinar. I will be presenting the technology aspect of the webinar because it's a new technology. It still needs to be explained, and then Nizar will dig deeper into the scientific questions that actually the technology can help you answer. Um, but I will explain a bit more what we are doing. But first, I still want to put you a little bit in context. Of course, Nizar will be more thorough in his explanations, but here is just a cartoon depiction of the tumor microenvironment. And we all know this is very populated by different kinds of cells. Now, our technology focuses on having a lot of markers on a sample, and you might question, why do I need that? Because until now I managed to get a lot of answers just by normal technology. So now it's a clear example of an experiment that was done actually by our R&D, and here you can see how different cells are known to be described by known markers, right? And just in that example alone, we use 44 markers to identify 30 different kind of cell types, and these are known markers. It's not anything about discovery. So you already see what is need to have such a high number of markers just to identify cell types. Now, as another example, and this comes from literature, there was a study showing that estrogen receptor positive versus estrogen receptor negative breast tumors shown actually almost 300 differently expressed markers. And this also hints at the need that we have to have so much more information when we are analyzing our samples. And for that, I'll be introducing you a little bit to our MACSima Imaging Platform. I will be changing terms a little bit. When I talk about platform, it's about the whole solution we offer. When I just say MACSima or MACSima Imaging System, I'm talking specifically about the instrument, but you all realize that it's a family affair that we have here. But if we had to describe the instrument in one sentence, we could say that the MACSima is an automated imaging system with which users can image more than hundreds of proteins on one sample in one experiment. Now, I will explain to you a bit how the technology works behind the MACSima. What happens is that we do basically cyclic staining in an automated manner. The machine will basically image your sample, stain it, and then erase the signal, and then do it again over and over again. And that way we can have on one sample hundreds of markers. And so far we have not seen the end of it. We do not know how many markers can be added in one sample. Our current record was actually 400 antibodies. And that way what you end up with, okay, it's not, yeah, there. So what you end up here is just to show you this is one sample, and this is a lot of different markers, as you can see there. Now, the way the technology works is so the cyclic, right, part, so we can do it over and over again. And how do we do that? And actually, it all comes to the possibility of erase the signal and then before restaining again. And for that, we offer actually two kinds of technology. So we use our recombinant antibodies are primarily conjugated with the fluorophore, and one of our first ways of doing it is via photobleaching. So we apply a high amount of energy to the fluorophore, so it's inactivated and will not show on the future cycles. Or we have or we have our releasable antibodies. So we have a big array of rheodialysis antibodies. With this technology, we will be able to release the fluorophores, and then they will not be shown again in the image. But we also have some portfolio of antibodies which are called the rhealyse antibodies, in which the whole antibody is actually removed from the sample. And the nice part of it is that you do not have to choose. You can use both mechanisms in one experiment if you need to. Now, when it comes to the antibodies and why am I focusing? So as I mentioned before, I will be talking about the platform as the whole solution we offer, and that's something that is very very specific on Miltenyi. So we do not only have a machine who applies the technology, but we have a lot more to offer. And one of these very important parts are the antibodies. So we have a vast range of antibodies who have been already validated for this technology alone. So we have validation already done in our portfolio. We have high quality recombinant antibodies. If you're familiar with Miltenyi, you know that this is something that we really hold dear to our arts, that we have high quality antibodies we offer, and recombinant antibodies is the best way to go. We have an extensive portfolio. We really have a lot of antigen antibodies, different clones, and even conjugates available to use in the technology, and we have different formats. So not only you can use antibody vials like you always use, preparing them before, but we also have predefined panels and very soon customizable panels that will even make the experience with the MACSima even more effortless than it already is. And just to mention that we offer this, but the MACSima is still an open platform, and it's still possible for users to use their own antibodies as long as they follow our rules, which is to be with bleachable fluorophores, primarily conjugated, and then it's how you wish, actually, quite flexible. Now, when we look at the system, why is it so effortless? It's because the machine has actually a lot of different components that make it easier. So we have a reagent stage that is very precise. That will move your samples or your reagent holders where it needs to be, either to be pipetted by the liquid handling or imaged by the microscope. And that way, you basically, after you insert your samples and your reagents, you can just go away and let the machine do the whole work. So it's really load your samples, antibodies, press play, and get on with your day. Rest is done by the machine itself. Now, and to complete our beautiful package, I want to mention that we also offer within this package an amazing analysis software. And if you are a little bit familiar with this kind of technologies, doing ultra high content imaging or spatial biology, you know that there is not so much to offer in this sector yet, and we offer something very, very unique with our image analysis software. It's a software that has been specifically made to analyze this kind of data set, so 100 of images, so you can easily display them. We have integrated segmentation, which is normally the first step of your analysis. We offer interactive gating, meaning that when you are selecting cells based on whatever they are in your image or on your plots, you can see them highlight everywhere. We also offer unbiased data analysis with k-means clustering, UMAP, and t-SNE. So if you are familiar with high content kind of analysis that can be, for example, genomics, you know that this is mainly used when you have so much information for one sample. And we offer multiple ways to plot your data in your analysis and do distance analysis also, because that's one of the biggest points of doing spatial analysis with everything, and a very comprehensible and easy to use workflow editor that allows you to really put there every step of your analysis and also recall it again. So this is a very complete package. It's not just for you to check your images, but you can really extract very important information from it. So just as a wrap up before I end the stage to my colleague Nisa, just a quick reminder of what we offer and what I mean exactly by the MACSima Imaging Platform. So we offer a very great instruments that makes it easy for you to make your experiment. But we also have a broad range of sample carriers. I didn't touch base too much on it, but we have sample carriers that allow you to analyze different kinds of samples, such as tissues, but also adherent cells. We have a vast range of antibodies that also are available in different formats. I didn't touch exactly on how the panels work, but basically instead of pipetting your mixture of antibodies in a big deep well plate, you could just put these panels in the machine and you don't even even have to pipette your antibodies. And of course, we close the gap by offering really comprehensive software. So not only to do your experiment planning, of course, but also a very nice and complete software for analysis. So I hope I didn't sound too much advertising. I really just wanted to shed light on what exactly we offer and how easy it is to use and to create all this amount of data that then my colleague will explain you a bit better what it is in the context of studying the tumor microenvironment. So Nizar, the floor is yours.
Okay, yeah, so now I will give you a brief introduction about the tumor microenvironment, yeah, and then I will present some application data generated by the MACSima in cancer and TME research. So what does, the tumor microenvironment mean? Yeah, so Telma explained it here a little bit. Yeah, so, well, the tumor microenvironment is a very busy environment, as you see it here, which in which many different components sit, yeah, such as cancer cells, immune cells. So these are immune cells that are resident in the tissue or infiltrating immune cells, yeah, and these are invaders. They come from outside the tumor, for example, from the blood stream. Yeah. So, and in the TME, we also have, extracellular matrix proteins and several secreted factors, yeah, as you see here. So in this cultural illustration, on the left, you observe a schematic of proposed mechanism of action of normalization strategies of the TME to improve immunotherapy. So combined administration of immunotherapy with a stroma and vascular normalization restores vascular functionality and elevates solid tumor stress, yeah, leading to improved tumor perfusion and oxygenation. So it's cueing tumor-associated macrophage polarization away from the immunosuppressive M2-like phenotype to the M1-like phenotype, yeah. So the M1-like phenotype macrophages, these are macrophages with pro-inflammatory phenotype, yeah. And there's the boost, yeah, these guys boost, for example, CD4 positive and CD8 positive T cells, and thus enhancing cancer killing. So the biological process occurring in the tumor microenvironment play an important role in tumor development and do affect the clinical outcomes of cancer patients. Yeah? So in this table here on the right, you see the effects of immune infiltrates on the prognosis of patients with cancer. And also in the table, you see the relevant CD8 positive T cells, yeah, tertiary lymphoid structure, Tregs, yeah, M macrophages, these are CD68 positive macrophages, and also more specifically macrophages with M1 or M2 subtype to overall survival, of different type of cancers. Yeah. So the presence of CD8 T cells, yeah, TLS, tertiary lymphoid destruction, as well as M1 macrophages in the TME is a good sign and indicates positive prognosis. Yeah, that's what you see here in green. However, the detection of M2 macrophages, yeah, CD68 macrophages, as well as Tregs in the most of the cases is associated with negative prognosis. Yeah, as you see here in red, in different type of cancers. So now researchers working in the cancer field do agree that it's very important to dissect, yeah, the tumor microenvironment to improve cancer therapy. Now, so let's have a look together how imaging technologies can help dissecting the tumor microenvironment. Yeah, so the question is, what is the importance of imaging in cancer and TME research? Yeah, well, since decades, imaging has been essential technique in cancer research and diagnosis. However, when we compare the present imaging technologies to the old school imaging technologies. Yeah. So we observe huge differences. So if we look at the imaging example from the old school, yeah, this is here shown here on the left. Yeah, this is the histological section from tumor stained with hematoxylin and leucine. Yeah, this is a known staining in the pathology field. Yeah, so pathologists, when they look at this section under microscopy, they can answer whether it's cancer or not. Yeah. So, but is this enough, for kind of understanding, the mechanism of cancer? Yeah. This is not enough. So, on the other hand, when we look, at present imaging technologies such as high content imaging using the mixed technology, yeah, this can allow you answering several questions. For example, what are the type of cells present in the tumor microenvironment, and also their spatial distribution. Yeah. So it is important to know the anatomical location of cellular interactions to better understand cancer mechanism. Yeah. Another thing is it also allows you, to know the type of biomarkers expressed. Yeah. So what type of biomarkers? Yeah. And also the features of the tumor microenvironment. Is it vascularized? Is it not? Yeah. And or does the tumor microenvironment consist of immune infiltrates? And where are, what are they? How do these immune cells, and other cells interact with the tumor cells and with each other? Yeah. So this is also important to understand this crosstalk between the immune cells, to each other, and between, the immune cells also to the tumor cell. Yeah. Yeah. So, and many other questions you can answer. So ultra high content imaging have great benefits for cancer and for TME research. So using single tissue section. So one tissue section, you go direction, ultra high content imaging. What can we do? You can stain hundreds of markers, which is indeed a benefit for analyzing TME complexity, yeah, with its large number of cell populations and structures, as Telma showed you before. Yeah, you can use ultra high content imaging to identify, for example, CAR T cell targets. And I will come to this to the next, in the next slide. So the MACSima, this instrument is a fully automated solution. You just need to insert your sample and let the instrument do the rest of the job for you. Good. So now, in my story today, I would like to stay a little bit, at the point of the identification of CAR T cell targets for the treatment of solid tumors. Yeah. As you might be aware, that CAR T cell therapy has shown much success in hematological malignancies. Yeah. But what is the picture in solid tumors? Yeah. In solid tumors, CAR T cell therapy has not shown any significant clinical results, in solid so far. Yeah. And so, but the question is why? Yeah. One of the main reasons hindering the success of CAR T cell therapies in solid tumors is the complexity of the tumor microenvironment. Yeah, we know the tumor microenvironment is a very complex niche. Yeah, for example, the presence of soluble suppressive factors in the tumor microenvironment, such as cytokines and suppressive immune cells, as I mentioned before, such as myeloid-derived suppressor cells. These are MDSCs and tumor-associated macrophages, TAMs, tumor-associated neutrophils, TANs. Yeah. Moreover, prevention of trafficking and the penetration of T cells into a solid tumor mass is another challenging factor. There are also other other factors that I'm not going to go through here. So therefore, researchers are currently busy trying to understand a better way that can lead to the success of CAR T cell therapy in solid tumor using, for example, modern imaging technologies that provides comprehensive overview of tumors and their microenvironment. And this brings me to the story actually I would like to share with you today. Yeah. So the story I would like to present you today has been recently published in scientific reports. Yeah. In this work, so with the help of the MACSima and its mixed technology, they analyze human glioblastoma, ovarian, and pancreatic carcinoma, and 16 healthy tissues. Yeah. So identifying the pair EPCAM and THY1 as a potential target for chimeric antigen receptor CAR T cell therapy for ovarian carcinoma. Yeah. So firstly, they tested this in ovarian carcinoma. Yeah. And by using an adapter CAR T cell approach, yeah, they were able to show selective killing of cells only if both markers are expressed. So let's dig a little bit, deeper on the story. Yeah. So this is an overview of the MACSima experiments they conducted. Yeah. So human glioblastoma, ovarian, and pancreatic carcinoma, and 16 healthy tissues were stained and imaged using the MACSima imaging system. And afterwards, the data they collected were analyzed, with the MaxIQ view software you heard about before. So now, to validate the performance of the technology on a tissue section, they performed a mixed analysis on a frozen section of a mouse spleen. Yeah. So in this experiment, they used 47 markers, targeting as many types of immune cells as possible, as you observe here on the image on the left. Yeah. So we see several immune cell markers, CD45, this is for a leukocyte marker, yeah, CD11B, myeloid cell marker, CD11C, it's a dendritic cell marker, CD4 T cell, CD4, it's a T cell marker, and so on. Yeah, so, and to generate the image you see here in the middle, as B, they performed image segmentation and determined the expression levels of the individual markers at the single cell level and grouped them into different clusters, using the k-means clustering. You also hear about it before, yeah? Then the clusters were subdivided into 4 main groups of cell types, yeah, as shown here, B cells in red, T cells in green, T cells in B cells, T cells in green, non-B cell and non-T cells, in blue or violet, as you see it, yeah, and the unlabeled cells in gray. Yeah. So, and on C, you observe the results of hierarchical clustering based on the main staining intensity values of individual cells and marker. And also you see a marker expression heat map, yeah? So of the various subpopulations. So uh, red means high expression, blue means, low expression, yeah? And also, you see the colored here on the left, you see the colored dot, indicates if a cluster consists mainly of cell types as classified here in B. So if they consist of, I don't know whether you see my mouse here, I'm pointing with the mouse, whether they consist of B cell, T cell, and so on. Yeah? So the conclusion here was that the mixed technology can differentiate multiple cell population in higher resolution and also provide, the spatial, distribution. Now, in the next step, they wanted to identify markers that are selectively expressed on solid tumor cells, but not in healthy tissue, to develop cell-based immunotherapy. In general, the identification of suitable targets, for CAR T cell therapy of solid tumors has been difficult, as most targets either do not cover a large enough percentage of tumor cells within a tumor and across different patients, or not exclusively expressed on tumor material, but also present on healthy cells. So therefore, they followed the strategy to first screen for targets which are broadly expressed on tumor material to allow for highest efficiency when, targeted by CAR T cells. For this, they used ultra-high-content imaging to screen several markers in different tumor samples, yeah, in glioblastoma, as you see here, pancreatic cancer and ovarian cancer, glioblastoma, pancreatic cancer, and ovarian cancer. In these images, the nuclei staining, DAPI is shown in white. The markers of interest are shown in green or red. Yeah. So, and this is an example of mixed analysis of 2 glioblastoma patient samples. yeah, showing the expression of GD2, EGFR, and CMET. GD2, EGFR, and CMET, as I said here in green, the mark of interest in green. And here, this is an example of pancreatic adenocarcinoma patient sample analysis showing the co-expression of EPCAM in red as a general epithelial and tumor cell marker with the potential pancreatic cancer marker candidates in green. Yeah. EPCAM in red, the potential in green. Yeah. And here, this is an example of, of ovarian carcinoma samples. Yeah. Sample 1 and sample 2 co-expression EPCAM in red and THY1 in green, while, in sample 3 expressing EPCAM abundantly, in the tumor cells, but, not THY1. Yeah. Next, they assessed the expression of target candidates in healthy human tissues, yeah, to make sure that the selected candidates do not cause toxicity on healthy tissue when used as CAR T cell therapy. Here, they observed the expression of the glioblastoma selected marker, yeah, GD2 in the brain. Yeah, this is here in the brain medulla. They also see they observed it in the cerebellum. It's not shown here. And a weak expression of this marker in healthy lung, pancreas, and ovary. Yeah, ovary, pancreas, and healthy uh, lung. Yes. So the EGFR was expressed in most healthy tissues, and CMET was expressed in pituitary gland, thyroid, lung, kidney, and heart. Yeah. This is thyroid, pituitary gland, thyroid are not shown here, but this EGFR and CMET is in the lung and in the expressed in the kidney. This is so in green and blue. So they then, when they found these guys are expressed also in the healthy tumors, yeah, they then made correlation analysis of healthy and glioblastoma samples with 3 marker pairs. Yeah, these are GD2 plus EGFR, GD2 and CMET, and EGFR and CMET all together. So, and based on this analysis, the combination of GD2 and EGFR appeared to be the most promising CAR T target candidates for the glioblastoma. So the combination of these two guys, not alone, the combination appeared to be the CAR T, the promising CAR T target candidates for glioblastoma. So then they use similar correlation for the other, tumor samples, approach, and they identified combination of, CDCP1 with TCPPAN8, yeah, as CAR T target candidates for pancreatic cancer. So for pancreatic cancer, they found TCPAN8, yeah, and CDCP1 in combination as potential targets. Yeah, and they also observe a significant correlation for cellular co-expression of EPCAM and THY1, and thus identified them as CAR-T target candidates for ovarian cancer. Yeah, so EPCAM and THY1, they were identified then together as potential candidate for ovarian cancer. So in summary, yeah, the mixed screening of multiple markers allowed them to identify marker pairs, yeah, that could potentially be used to specifically eliminate tumor cells while not harming non-tumor cells in CAR T cell therapy approach. So this is the quantification of the marker expression, yeah, in healthy samples as compared to cancer samples. I talked to you about it in the previous slide. Yeah, so confirming that some of these selected target candidates are expressed in healthy tissue as well as in cancer tissues. And as I mentioned in the previous slide, the correlation analysis allowed them to identify target pairs as promising CAR T cell therapy candidates. Yeah, as you observe here, some of the markers were expressed in the tumor tissue as well as in the healthy tissue, and that's why the pair was the solution. Yeah. So now, after identifying marker pairs as potential CAR T cell therapy candidates for the different cancers, they tested on one of the identified pairs. Yeah, so they just tested one of the identified pairs, which they identified for ovarian cancer, not for glioblastoma and pancreatic adenocarcinoma. They went for the ovarian cancer, Yeah, so they generated ovarian cancer cell co-expression EPCAM and THY1 in combination with the green fluorescence protein GFP. Yeah, so when they tested CAR T cell functionality in vitro using antibiotin CAR T cell in combination with biotinylated, EPCAM and biotinylated THY1 antibodies, they observed effective CAR T cell killing of target tumor cells. Yeah, you observe it here in yellow. Yeah, indicated by the decreased GFP signal. And this is actually was only the case when adapter CAR T cells were, incubated with EPCAM and THY1 together, at the same time. So now this is a summary of the paper findings. Yeah. So the MACSima and its mixed technology helped identifying EPCAM and THY1 as potential targets for CAR T cell therapy for ovarian carcinoma. And using adapter CAR T cell approach, they show selective killing of cells only if both markers EPCAM and THY1 are expressed. Yeah, they also show the MACSima and the mix as ideal, technology for parametric analysis of human tumor samples. Yeah, and they say that the mix represent a new high-content microscopy methodology widely applicable for personalized medicine. Yeah. So the paper I presented you today was, an example showing the benefits of the MACSima and its mixed technology for characterizing TME and cancer, spatial biology. So there are other nice publications available, online showing how ultra high content imaging can bring cancer research to the next level. Yeah. So I recommend you to have a look at these publications. For example, these ones shown here on the screen. Now, I would like to sum up my talk. Yeah. So deep characterization of the TME components, including spatial distribution and the ability to analyze, high number of markers. Yeah, this can be possible using the MACSima and mixed technology. So the MACSima allow you identify tumor-specific target candidates for therapy, for example, CAR T targets. Yeah, also allow you identification of complex TME processes, cellular interactions, cellular infiltration, and immune responses. Yeah, we have extensive validated antibody portfolio, as Telma presented you for direct usage on the sample. Uh, and of course, MaxIQ View image analysis software, yeah, which it comes in, or accompanies the MACSima, yeah, allow easy analysis of complex TME data, because you generate this a lots of data. Of course, you need to have a better way, or an easy way to analyze them. Yeah, the MaxIQ View is your friend. Yeah, and also obtain huge scientific data. You can obtain huge scientific data from limited sample material. Easy, automated, you don't need to worry about any, staining and or images. The instrument is automated. Yeah. So you insert your sample and it does the work. So, and most importantly, yeah, our internal experts are ready to guide you throughout the entire ultra high content imaging work. So we will not leave you alone. We will accompany you from A to Z. And here, actually, I finished my part.